Hello and welcome to the Made Easy series, episode on genetic engineering. I was going to add CRISPR into the mix, but Bosamon Science did a better job than I could ever do, so I'll just recommend that video to you in the description below. In this episode we'll cover the use of the particle bombardment technique and the Agrobacterium technique. Both methods involve the use of tissue culture. I go into more detail in an episode on my BitChute channel regarding tissue culture history. Again, linked below. But to make a long story short, tissue culture is a method where whole plants are grown from plant cell cultures, produced on petri dishes. Now on to the video. We'll first cover the particle bombardment method of genetic engineering. In this method, materials like tungsten have the genes added onto it. These particles are then bombarded onto a plant cell culture through tissue culture. The force of the tungsten forces the DNA into The benefit of this method is that you can use this method on any plant material, and it can add up to 3 genes into the plant using this method. There are problems with this method, however. Multiple copies of that DNA could be added to the plant material, so one cell could have multiple copies of the same gene, causing trouble for the plant. Another problem is that where it winds up would be completely random, and if it doesn't wind up in the right spot, it could do nothing to the plant or be harmful. The first known Roundup Ready soybean was made through this method though. Next we'll cover Agrobacterium mediated genetic engineering. This species of bacteria carries a circular piece of DNA called a plasmid. This species inserts its plasmid DNA into the plant cells in order to induce mutations in the plant to make it more suitable for its growth, creating strange growths as seen on this image. In this method, the bacteria are modified by replacing the plasmid with the gene de jure and the marker gene, which we'll explain a little bit later. Following that, the process seen on the screen happens. The bacteria get introduced to a plant cell culture of the crop you want to genetically modify. The bacteria, upon being added, inject the gene-marker gene combination into the plant cell. This typically results in the gene and the marker gene being inserted into the right area of the genome. A benefit of this practice includes better fertility from the plants themselves and a higher rate of successes. But a drawback is, to prevent the bacteria from destroying the cell culture, antibiotics need to be used after a period of time to end the colonization process. Another drawback is that it only works on certain dicot. Regardless of what method is used, you still need to track the genes and make sure they're in the right spot. With the Agrobacterium gene method, the researchers use marker genes. These genes become active as soon as the regular gene becomes active due to how closely connected they are. Such genes include the gene from a jellyfish that causes the plants to fluoresce when exposed to a certain type of light, or the Roundup Ready gene, which allows the plant to survive Roundup. By either testing out to see if the cell culture survives Roundup or if the plant cells fluoresce, one can tell if the gene addition to the plant cell culture has been successful. Following the completion of this project, and if the gene addition to the plant cell culture was successful, the plants are then grown up into fully grown plants to be used for breeding purposes, to produce a genetically modified cultivar. Well, that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.